This is our next talk in the series that is celebrating undergraduate research. Because this is Utah State University's year of undergraduate research. So we have a variety of events going on throughout campus for the whole 2025, so stay tuned. My name is Brynja Kohler. I'm the chair of the Science Unwrapped committee and also the associate dean in the College of Science for undergraduate programs and services. How many of you have been to Science Unwrapped before? Please raise your hand. Wow, awesome. There's almost no newcomers, but for the newcomers, let me just tell you that we'll begin with a talk, and after the talk, we'll have time for questions. We'll do a question and answer session. We're gonna take a little break between the end of the talk and the question and answer session. So if you wanted to go right out, you can excuse yourself quietly and go out. But if you want to stay for the question and answer session, then just come up forward and we'll get that going shortly after the talk. So there are refreshments and hands-on learning activities after the talk in the atrium. And we have lots of volunteer groups representing different clubs. And also community organizations that are here with lots of cool science learning activities. So be sure to check those out. I also want to mention we have 2 undergraduate students who are presenting research posters this evening. Chase Pyra will be explaining why carbon monoxide might not always be bad for your health. And Ellie Hartman has a poster about snake behavior after they've been challenged with, immune challenges. So be sure to visit those students and check out their undergraduate research posters. So for our talk this evening, we're very excited to welcome tonight's speakers, USU Biological Engineers Elizabeth Vargis and Sophia Hessami. Dr. Vargis is an associate professor in the Department of Biological Engineering and leads a robust research lab focused on the study of nanoscale cellular interactions and improving models for studies of human diseases. She and her students are using biomedical engineering to find ways to prevent or slow the progression of many conditions, including diabetes, cancer, and other illnesses. Sophia Hessami, who accompanies Dr. Vargis this evening, is among those students. Sophia is a USU undergraduate research fellow, and at this time last year, we celebrated as she was named a 2024 Goldwater Scholar. The Goldwater Scholar program is one of the most prestigious and competitive academic programs in the nation for undergraduates studying science, engineering, and mathematics. Tonight, Sophia will share her fascinating journey in undergraduate research as she and Dr. Vargis discuss their research on a common eye disease that affects many people around the world, including people you may know here in Cache Valley. Let's welcome Dr. Elizabeth Vargis and Sophia Hessami.
When I started college, I pretty much knew immediately what my major needed to be. My mom was a microbiologist, so she worked in a hospital lab helping doctors figure out what type of diseases people had. And my dad was an engineer, a mechanical engineer, so he worked at a lot of manufacturing companies. And so I knew what my major had to be. I didn't want to pick any sides or make anybody angry in my home, so I chose bioengineering. And so they could both appreciate it. And a bioengineer is kind of a applied biologist, so we use biology to study disease. We use biology to make artificial limbs or new sources of fuels or different medicines. And I loved being in college, and it was pretty easy to figure out what type of classes to take because there were counselors and advisors and faculty members to help me out. But college is super fun, and you don't just have to take a bunch of classes. You can do things outside of the classroom. So I joined an a cappella group. I worked for the California Department of Public Health, and one day I took a chance on becoming an undergraduate researcher by signing up to work in a cancer biology lab on campus. And from that one decision, that, steamrolled the other decision. So I started to decide and think about going to apply for a PhD. I moved to Tennessee, and all of those decisions led me to this faculty position and the opportunity to work with amazing students like Sophia.
My name is Sophia Hessami. I am an undergraduate student. I grew up in Washington State and moved to Logan, Utah 4 years ago, August of 2021, to start my schooling in biological engineering. I wanted to start with just a little bit of background in terms of why I chose biological engineering. So Dr. Vargis mentioned that it really is an applied biology. And growing up through middle and high school, my favorite subjects were always math and science. So when I stumbled upon a major that combined those two, I was pretty stoked to kind of further explore and commit to studying this. So with that, I want to get started, and we are going to poll the crowd a little bit here. So by a show of hands, if you had to give up one of your 5 senses, which would it be? Who would give up their sense of taste? Okay. What about hearing? Sight? Not a whole lot of takers on that one. Smell? Wow. And what about touch? Okay, so smell knocked it out of the water there. I'm pleased to see that most of you chose to keep your sense of sight, and you guys set me up perfectly for the rest of this talk. So we're going to be talking about vision, we're going to be talking a little bit more about our eyes, and then we'll dive into the disease, age-related macular degeneration, and talk more about that and how we research it in our lab.
Hi. Is this working now? Yep. Okay. So we're just going to start with the basics of how vision research, or how the eye works. And so you have light that enters through the front of our eyes, through the cornea. It travels through the small hole in the very center, our pupil. It then travels through the lens to be focused on the back of the eye, which is called the retina. The retina is a really special part of our body. It's light sensitive, and it's what sends the signals that hits it. It sends it through the back of the eye, through that tube. It sends all those signals up to the brain. The brain rearranges everything and unscrambles it all, and then that's how we can interpret all the different light and images that we see. And the eye is just really, really, really cool, okay? So it's such a super specialized part of our body, but it also has every single component that we find throughout. So it has a ton of muscles, right? So you can move your eyes back and forth, but it also just responds to the amount of light we have. So the pupil just adjusts accordingly if there's too much light or too little light. It has its own blood supply, so it has 3 or 4 different ways to get blood into the eye, and that provides all the nutrients that the eye needs because it's really energetically active, so it needs a bunch of food sources. And it also gets rid of any waste. It maintains the pressure and the temperature all through its own little blood supply. And the eye also has something called immune privilege. It doesn't fight bacteria and viruses like the rest of our body does. The eye does something just a little bit different, so it kind of has its own immune system. So all of that is just located in our little eyes. And in general, we're pretty good at recognizing when people have a little bit of a vision loss and need a little bit of help. So in this room, for example, how many people here wear glasses or contact lenses? Right? And so this is pretty much a normal part of what we go through. You get your eyes checked, your vision checked in school. And as an adult, seeing an ophthalmologist or going to the eye doctor should be a part of our annual doctor visits. And wearing glasses and contacts can be kind of annoying, but it's a pretty easy way to fix any sort of nearsighted or farsightedness or even some astigmatism. But some eye diseases are a lot more serious, and something as simple as glasses and contacts won't fix those problems.
And so one of those diseases is age-related macular degeneration. So just to give you guys a picture of how many people are affected by this disease, in the US alone, there's roughly 20 million people that are currently diagnosed and living with age-related macular degeneration. And if we zoom out worldwide, that number is closer to 200 million. So it's a very common disease. Before I dive into that, in terms of how it affects your vision, we start to have kind of a blurred, distorted, warped, and when the disease is far enough progressed, kind of black spots in the center of our vision. In terms of who is affected by this disease, like I mentioned, people over the age of 40 are largely affected by it, but there's a lot of other risk factors that go into it. Some of these risk factors are things that we can't control, so age and family history, tough to, control that one. And then there are also some things that we have a little bit more control over. So smoking, our diet, and then also exposure to sunlight will have impacts. And so like Dr. Vargis mentioned, there are some ways that we can be diagnosed of this disease. And a really important thing to work towards diagnosing and making sure your eyes are healthy is, like she said, going to those yearly checkups with an ophthalmologist. And so what an ophthalmologist does, one of the many things that they do during these eye exams, is some retinal imaging. So this machine here takes a look through your pupil at the back of your eye, and it takes an image of your retina, and it allows these doctors to see whether or not things are looking healthy. So in a healthy eye, this is what the back of your retina looks like. So that kind of lightish pink spot in the middle, that is our macula. So the macula is in charge of all of our central vision and makes sure that the images we see are really sharp and clear. And when everything is normal, it looks something like this, and our vision is going to be nice and clear. Now with AMD in the early stages, we might start to have this accumulation of those kind of light white spots that you see towards the middle of your macula. What those spots are an accumulation of waste products that haven't been properly disposed of, and that can kick off kind of this unusual fuzzy or distorted vision that you see down below. And then finally, as things progress further, either if you're not diagnosed early enough, or there's no adequate treatment for you, then it will progress and you will start to have the actual degeneration of your macula. Or another way to think about that is your macula starts to break down. So within your macula, there are some really key cells that help us see. And so when they're broken down, that's how we start to have that blind spot that's pretty traditional when we think about age-related macular degeneration. And that's when that vision loss gets kicked off.
So zooming in a little bit closer, if you look at the macula, that's that center spot, like Sophia said, it's about 5 millimeters large, which is like the size of a pencil eraser. And that's where all the good stuff happens. You can see shapes and people's faces. It's important. Our central vision is super important. So if we zoom in right below that, what we can see are another set of really specialized cell layers. So we, of course, have our photoreceptors. Those are our rods and our cones. They see lights, shape, darkness, all the differences that we see in different times of day. And then right below them are the retinal pigment epithelial layer. This cell type is really just there to support those photoreceptors. I like to think of this layer as those people on like sports teams who are always passing out the Gatorade bottles for like Caitlin Clark, or like making sure it's the right tennis ball and towel for Coco Gauff, right? That's what the retinal pigment epithelial cells are doing. They're just there to support the photoreceptors, because the photoreceptors are the big deal in the retina, okay? And so we need the retinal pigment epithelial cells are what takes care of everything else. Then there's this really thin layer underneath there, which really just serves to separate the blood vessels from everything else in the retina. That cell, the Bruch's membrane layer, has no cells in it. It's just really thin and just creates a barrier. And the choroid is the one set of retinal blood vessels that, again, provide those nutrients for the back of the eye. It takes away waste, and it also provides food for these cells that are involved. So like Sophia mentioned, when people have age-related macular degeneration, this stuff can't really be fixed by glasses or contact lens, and there's just something that's going wrong between these layers. So that first version, which had the drusen, the little deposits that were forming, that usually happens right below the retinal pigment epithelial layer and Bruch's membrane, and they're kind of fatty deposits, they're mineralized, and that's the initial stages of macular degeneration. And then when things get really bad and you have these blood vessels growing and you have really severe vision loss, this is what's happening. You've lost some of those retinal pigmented epithelial cells, and then you've lost photoreceptors. And as you know now, the photoreceptors are what allows us to see. So then if we lose those, that is the indication that we have severe vision loss. So we've told you that a lot of people are diagnosed with age-related macular degeneration. It's not that hard to diagnose people, so that's not really the problem. The problem is there are still really limited options. So if you have that version where there's deposits underneath your retinal pigmented epithelial layer, your treatment options are none. You can take this specialized vitamin, which you can get at any store. I took this from Costco's website. So that can help you prevent it if you have the familial history. You could take these if your doctor recommends it. If you have the worst version, where you have blood vessels flowing through your eyes and you have pretty severe vision loss, your options are to have a laser treatment every time those blood vessels are in the back of your eye, or monthly injections into your eyeball. Right. I could hear from the gasp that that's probably not a great option, but this is vitamins and two really pretty invasive treatment options. So I don't know about you, but I feel like there's a better way. And so before we get into how I think that there are alternatives for finding new treatments and ways to prevent this disease, how do you all think that scientists study disease? So how could we come up with new treatments? What do we use? Yes? So you mentioned both humans that already have the disease and some sort of animal that we either give the disease to or they catch the disease. Yep. Yes?
You could look at similar diseases and look at what affects those and then try them out on the new disease.
So if there's some similarities, another option would be if there were similar diseases, so if there were other deposits or blood vessels that were coming through, maybe we could find new treatments by looking at those. Anything else? Yeah, so I just put together like these kind of 3 stages. So we can study disease by looking at just those cells as they're growing. So one layer, one at a time. Kind of can do this in the lab. We have the setup in my own space here at Utah State. Then, yeah, we have to move on to some sort of animals. So it could be literal guinea pigs. It could be mice, rats. And we either give them the disease or at least we study how vision happens normally, and maybe we expose them to sunlight or cigarettes and see what happens. Or we can go all the way to humans, either normal humans or humans that have age-related macular degeneration, and then we figure out what went wrong, what was their genetic history, and those are the big components of our options when we're trying to find treatments for diseases. So here at Utah State, in my group, we don't touch these. We really focus on this first step. But most of us see this step as a bunch of cells growing on thick pieces of plastic. And I don't know about you, but I feel like that doesn't describe the human body very well. And so what I try to do in my group is to make this process better. So I move away from plastic dishes. I'm try to use cells that are actually more realistic, more like the cells that we have in our body. And I stay within this realm to make the next step, to make the results of that first step easier for the next step. So that if we can trust those initial results, then maybe everything else will work out better. And so we're in this realm, but we're just trying to do things a little bit better and a little bit different.
And so this is where I come in. As an undergraduate researcher, I have a lot going on. I guess I want to start by saying I do attend classes. Undergraduate students have 3 to 5 classes each semester. So I go to those, I do my homework, I have my projects. But what I really love doing is research. So with any extra time I have, I'm in the lab and working on research. So the fun thing for me about research is that we're trying to discover new things. And that often starts with literature review. So literature review is important for a few reasons. One, as an undergraduate who came to USU not having any knowledge about age-related macular degeneration, it was important for me to build a baseline understanding of what the current status of this disease is and what other scientists have discovered about it. So that's the first thing, getting a foundation of knowledge, figuring out what other scientists know. And then the next thing is identifying gaps in the literature. So once you identify what isn't known about, in this case, age-related macular degeneration, that can help inform what future experiments need to be done. So that's kind of my next step in my week in a life. I start to plan out experiments. So for me, what that looks like and what I found when I did my literature review was that there isn't a lot of information in terms of how sunlight or UV exposure affects our retinal cells and how that can contribute to the development of age-related macular degeneration later in life. And so I've read my literature, I've planned that I want to look at UV and how it affects retinal cells, and my next step is now getting my hands on retinal cells and moving forward. So before we move on to this next picture, there's going to be some pictures of real pig eyes that we dissect in lab. So if anybody feels a little bit squeamish about that, now's your chance to close your eyes, and I'll let you know when they pass away from the screen. Whatever.
Okay, last chance.
Okay, so we need to collect these retinal cells. And the way that our lab does it is we have a local butcher that we go to once a week, and we're able to get pig eyes from them. So that far left image is what they look like when we get them. There's a little bit of excess tissue and muscle surrounding it. So we clean those up, and then in that third image, you'll see that we start to dissect them or cut them open. And so that dissection process, we're essentially getting rid of all of the stuff that we don't need. Which they're important parts of the eye, but I just don't need them for what I'm doing. So the cornea, your sclera, your lens, all of the fluid that's inside of your eye, all of that gets tossed. And we're left with this cup where we're able to access the retinal cells in the back of our eye. So once we kind of scoop those cells out, we then can put them in something pretty similar to a Petri dish. Something similar to a Petri dish. We call them well plates or transwells, and that is just a space that these cells can live and thrive outside of the pig's eyes. And so that is another big part of what my week goes to, is taking care of these cells and making sure that they're all good to go so that I can actually do experiments on them. And so that second image is what these retinal pigment epithelial cells look like. So you'll notice there's a really nice kind of cobblestone, thanks Dr. Vargis. There's a really nice kind of cobblestone layer look to them, and that's exactly what they should look like. And then in addition to that, once we have those cells into a dish that we can not raise them in, but take care of them in, we have to take care of them, and part of that process is feeding them. So just like you and I, these cells need to be fed about twice a week. And that is all done in a biosafety cabinet. So a biosafety cabinet is a really cool environment that essentially just keeps the cells nice and clean and happy. It has filtered air, and it makes sure that any of my germs that I have don't get onto the cells and get them all gross and icky. And then that food that I was talking about, that's cell media. So the food is simply just a liquid that has all of the key vitamins, minerals, and proteins that the cells need to live and thrive. And so once my cells are all good to go, they're growing, they're happy, they're healthy, then I can start looking at actual experimentation with them. And so my next step is I identified that the gap in the research was whether or not, or how UV affects these retinal cells. And so I first had to figure out, okay, how am I going to expose these cells to UV? I can't just put them outside and let them hang out. That's not going to work because I need to know exactly how much UV is actually hitting these cells. So I talked with Dr. Vargis, and she said, well, I don't have any sort of UV source that we can use. So we reached out to someone in the physics department that's actually here tonight, Dr. J.R. Dennison, who does a lot of research with UV. And he was kind enough to let me borrow his UV source. Looks kind of like an oven. It's about the size of a microwave. It just has UV bulbs that emits UV light onto my cells, and I can know for sure exactly how much is hitting those cells. Okay, so now I have my cells. Now I have a way to introduce UV. Then my next steps is figuring out how the cells are affected by this UV. So there's lots and lots of ways that scientists and researchers can figure out how their cells are being affected by any given experiment. This is just one way. And honestly, it's kind of one of my favorite ways because I think the pictures end up being really pretty. But here we're able to see on the left is what our retinal cells look like when they're healthy and happy. So you'll notice that green outline outlines the edge of every cell, and we have that really nice, consistent cobblestone look. And then if we move to the image on the right, these are cells that have been stressed and have been exposed to a stressor. And what happens there is you'll notice that one, that cobblestone layer looks a little disrupted. We have some gaps, and the outlines of the cells just don't look as clean and crisp as we have on the left. So once I have some results, then I move forward to kind of packaging everything up and presenting it at our weekly lab meetings so that I can check in with graduate students and Dr. Vargis to see how things are going. And that is a really valuable opportunity for me because I'm able to bounce ideas back and forth on people that have a lot more experience than I do. And I'm also just able to get a little bit extra mentorship and support, which is very nice as an undergraduate researcher.
So Sophia is a student in a long line of students who've been able to work on understanding and preventing vision loss here at Utah State. So I'm just going to walk through a couple of other projects that we've had. So we started with the worst case scenario, so that one where the blood vessels are coming in and the retinal pigmented cells are dying and the photoreceptors are dying off. And we thought, could we make those lab experiments a little more realistic? Could they actually mimic what's going on? So we started by that same layer of those retinal pigmented epithelial cells from the pigs, and we said, okay, what if we could control where the cells were growing? So we 3D printed a mold, we added this jello-like stuff on top of it that ended up hardening, and we had it basically be a stencil. And so we could put this stencil, it is this one over here. These are just like holes in the piece of jello. And we could put that on a plate with all the cells, pull the stencil off, and then have these beautiful circular patterns where all the cells were growing in there. And basically, that's how we could say, how do those cells survive once their neighbors have died off? But this isn't how it looks in our body. Nothing is perfectly circular. That's not what's really going on. So a new student came on and she said, this isn't what it looks like. Why are we doing all this work? So let me try something different. So she created stencils that look a little bit wonkier, right? There's no clear edges. It's not a perfect circle. She took a bunch of images of what damaged macula looked like and kind of morphed them all together, printed this out using the same 3D printer, the same setup in the engineering department, and has the same stencil, pulls that off, and then is left with, these are what they look like. They're smaller than quarters, and is left with this area where the cells start off pretty normal, but then on the edges where they're trying to respond to losing their neighbor, they have lost, again, that cobblestone morphology. They don't look, this is how they look healthy, and as they get to the edge, they don't look like that anymore. So when you have the initial stages of the disease where the blood vessels are coming in and killing off your cells, the cells that are left are not normal anymore. They're responding by acting abnormally, by losing a lot of their function, and this just continues. So then we said, okay, we can do this pretty good. Let's try this one. What happens when we have a little bit of pressure from these new deposits? And so again, a student came in and I said, could you make this in the lab? We're engineers. Let's do something like this so that we can make our models mimic what's happening in humans. And so we started off by pushing things on cells, things that we could find in the lab. So we had pipette tips, we had tops of pens, other random objects. And what we found is if we pushed on layers of cells on our own, we often broke through the layers. So then we were really smart and we went to the thrift store and we bought a sewing machine and we put those pen tips and pipette tips onto the top because it was our fault. We were just too strong. So we were going to use something that we could control. And we still busted through those cell layers. And so you live and breathe this at a certain point. And so I didn't know how to solve this. We tried all sorts of things, and it was the cotton swab wasn't hard enough. And so, I thought maybe we could just end it on this. And, this is just like an extreme version of macular degeneration, right? This is like when your retina tears from the back of your eye. Somebody cares about that. But then on a Saturday, I was with my daughter, who's in the back over there, and we were getting a pedicure. And after the pedicure, they offered the nail art. And they brought out the tools that have the little ball on the end in order to make flowers. And I thought, that's the solution. And so I brought in another student, and I bought a bunch of manicure and pedicure tools. So when I get audited, you all know that I wasn't using it. We did not use it. My daughter and I have never seen those in our house for anything else. It's used for science. We put it on the end of over here. And so these are manicure tools, okay? These are a layer of cells. We can control this. We just added it to some other pieces of equipment in our lab so we can control the amount of force that we're putting in. Because again, we just want to see what happens to the cells that are remaining. The deposits are already there. Are the cells now functioning appropriately? We found that the answer is no. So sometimes we find things that other people have already found. Sometimes we corroborate those human studies that you were talking about. Sometimes we find new things, and possibly that would give us more insight because we're looking at things from a different point of view. And so this is a cool story, but sometimes I have students who try these little side quests and do things because of reasons and find totally unexpected results. So I had a student, and he was going to a conference and then on vacation. And so you remember Sophia said that our cells have to be fed every 2 to 3 days. He was going on vacation for 2 weeks. He didn't want to feed them, so he decided to just let it be. So here's what they looked like at 2 weeks. He let them continue growing. So here's what they looked like at 1 month. And then he thought, they didn't really die, so I'm just going to feed them like twice a month. And after 4 months, they look like that. Does anybody see a big difference? I didn't. Honestly, to me, the 4 months looks the healthiest. So the cells that were fed the least seem to be the healthiest. So, whoops. This was unexpected. And so this is a whole new idea. What are the cells doing? What are they eating? Because we've been feeding them every 2 to 3 days. So there must be some other pathway that these cells are able to use, and maybe that is the whole idea, because maybe they have to give all their food to the photoreceptors. We don't know, we're not using those in our cells. So this is a whole new line of research because a student decided to maybe be lazy, maybe try something new, maybe just see what happens. And I welcome that in my lab because this is neat.
And so, in terms of unexpected results and just how that impacts me as a researcher, there's a lot of ups and downs in research. So I mentioned how, I do my literature review, I identify a gap in research, and that informs my future experiments. And so I wanted to talk a little bit more about that gap in the literature. So I'm exposing these cells to UV, right? And there are other people that have done similar work, and that work has been done on these cells on the left. So on the left, we have ARPE19s, which are retinal cells, but they aren't looking very similar to the cells that are actually in our body. And that's a really important distinction. So these ARPE19s, you'll notice a few key differences. One, they're a little bit more elongated than the cells that I work with on the right. They also don't form that nice cobblestone layer that we have. And then most importantly, they don't have that dark pigmentation that we see with the cells on the right. And that dark pigmentation has, for better or for worse, led to some complications in my research. So this pigmentation is our retinal cells' natural sunscreen. It helps protect our retinal cells from UV and sunlight so that when we go outside and hopefully we're not staring at the sun, but sometimes it happens, we don't immediately go blind, which is great for us. Not great for my research though, when I'm exposing them to UV, expecting them to die, and they're not quite dying. And so that kind of goes into the ups and downs of research. There, at times, are failures, and there's a lot of trial and error. And I think when I was in my first and second year as an undergraduate student, those failures felt a little bit personal, I guess. It felt like I was doing something wrong. But luckily, I stuck with it. And at this point, I have been able to recognize the importance of failure as a researcher. There have been no great scientific discoveries without a little bit of trial and error and failure along the way. And I feel really fortunate to be in a lab that encourages failure. And Dr. Vargis and the grad students in the lab have all been really great in encouraging me to continue pushing and continue exploring new avenues in research. And so, yeah, I feel really grateful for my opportunity as an undergraduate student to have this hands-on access to learning and to have the opportunity to fail a little bit along the way.
Yeah, and so just to wrap things up, we really support any sort of student who wants to do any hands-on research. We are a supportive environment of failures and successes. And so I just put together some lab photos over the years. There's one here. So most of these are, college student, but this person right next to me, she was in high school. And so she came to our lab and she volunteered and came to all of our lab meetings. And, we do work in a basement and there are no windows. And so sometimes it's tough, but we have a lot of fun. And we have collaborators who have really beautiful labs with views of all of Cache Valley. And you can always go work with them as well. I think that none of this would obviously be possible with a wide range of funding sources. And so the ones at the bottom, the Retina Research Foundation, the Knights Templar Eye Foundation, and the Bright Focus Foundation are all private. And so these are philanthropy, rich people putting together some money to fund certain causes. But the ones all along the corners are federal and state funding. And so we've been really grateful to have that funding to be able to support projects and side quests from really good students like Sophia. And so, to wrap up, we're just doing our best to try to prevent vision loss, but of course you have your own role in this, so you do have to have a good diet, you have to exercise. Yes, carrots are important, so there's beta-carotene in carrots that becomes vitamin A, which is really useful for the cells in your eyes. There's even something called lutein, which literally helps your eyes stay pigmented and apparently will protect you from all sorts of sunlight exposure and UV light exposure. You need to continue, wearing those sunglasses, even though we said UV light, may or may not hurt. Wear them, especially at the beach. Go to the doctor, take your vitamins, the general ones. You don't have to take the special ones. Get your vaccines. And with that, between all of us working on this, I think we can prevent some of the really dire cases of vision loss. But I appreciate all of your time. We appreciate being invited here. And please come work with us.
All right, great. So let's go ahead and get started with questions. You'd like to start?
You said to take general vitamins, but you don't have to take special vitamins. What does the research show as far as like which particular vitamins really help prevent macular degeneration?
So if you have I'm not a doctor. I'm not an ophthalmologist. If you have like a family history of AMD, most likely at a certain age they'll suggest that you take that specialized. It's called ARIDS 2, and it's available everywhere. But that's only if they believe that you might have some of those deposits forming. And so maybe when you go to your annual doctor's appointment, they'll be like, okay, we see that happening. But otherwise, any sort of adult multivitamin would be okay.
Okay. Go outside question?
Oh, we left.
Come on, someone must have a question.
We provide the scientists, you provide the questions.
Yeah, Sophia, I was wondering, when you were doing the UV light exposures in your experiment, compared to when you go outside and it's sunny or whatever, how much UV radiation were you actually exposing them to?
So the trials that I've done so far, disclaimer, I still haven't found a UV level that has actually imposed death. So that was we're still working through that right now. The UV that I'm exposing is a particular wavelength of UV. So on Earth's surface, the UV I'm exposing is about half the intensity, but in terms of time frame, it's a lot. So those cells will sit in there for up to 40 minutes, and that is, I think, I've crunched the numbers, I think it's equivalent to a year of being exposed to the sun constantly, which that's not what is actually happening in real life. For me, I'm in the basement with no windows, so.
You're welcome.
Thank you.
We're supposed to repeat the question.
So my son has this really cool pen that he got at book fair, and it's a secret message pen, UV light pen. The dude likes to shine it in his eyes. And it says on the package, don't shine it in your eyes. And I took it away from him.
Good.
Yeah, I wouldn't recommend that. Our eyes are good at filtering out UV, so obviously the studies I'm doing are just with those retinal cells, but in real people, we have a lens and that absorbs the vast majority of UV. So he'll be okay, but I think good call on taking the pen away.
No joke. And his grandma gets those shots too, so he already has family history of it, and I'm like, he got mad at me. Anyways. I have another question.
So when you run a lab like yours, how often do people have to be in it? Does it run all weekend? What does it actually look like to run that lab in terms of people hours?
So it depends. Sometimes we'll have experiments going over the weekend. I'll be in there tomorrow, actually. In terms of general lab upkeep, so like with feeding the cells, they need to be fed around twice a week. Usually it's kind of up to each individual person. Whoever's cells they are, they're kind of in charge of making sure those cells get fed. If you're out of town, you can do what the PhD student did and just let them sit and see what happens. But usually you either have to check in with a grad student and see if they'll take care of your things, or just plan your experiments efficiently and make sure that you'll be there when you need to be there. In terms of like my time commitment, I probably spend anywhere from 10 to 20 hours a week doing lab work. Most of that is in the lab, but then when I'm gathering data, a lot of that is like I'm at home crunching numbers on my computer. So.
So when you get those cells from the pig eyes, how do you separate one layer from the next, the pigment cells from the rods and cones?
That's a really good question. Yeah, so we start out by first slicing them open and getting out all the jelly and stuff that gives our eye its structure. And then there's kind of just one more layer that needs to be removed before we're actually at the retinal pigment epithelial. And that layer we truly can just peel right off. So we put a really cold liquid on top and it kind of helps with the peeling process. And then we just get in there with tweezers and pull it off. To get the RPE cells off, we put a special solution that helps them detach from the eye. Most of the time that is successful and we don't have any other contaminating cell types. Sometimes we do have other cell types in and then we're not able to use it for these experiments because we want to make sure we just have those retinal cells.
So, follow-up question. Do they replicate or do you only get the number of cells you get from that eye and that's it?
That's a really good question too. So these particular cells are postmitotic, which means even the cells in our eyes, that's just what we have. They don't divide any further. Whatever we're born with, that's what we're stuck with. For these experiments, once we pull them out of the eye and we put them in whatever dish we're growing them in, you can force them to divide. It's not ideal for a few reasons. One, since that's not what happens in our eye, it makes things less mimetic. And two, that pigmentation that we talked about, the amount of pigmentation you have that's just set. So if they start dividing, you divide your pigmentation in half. If you divide them a third time, then they're cut in half again. So you start to lose that sunscreen for your eyes pretty quickly, and it just makes them less mimetic.
Do you happen to know if reflected UV light is as bad as regular UV light? So say I, sometimes stare out my kitchen window and there's this white surface there and I suddenly realize, oh man, I've been staring at this for a while. Do I need to worry about that?
I'm not 100% sure. What I do know is that our eyes do filter out a lot of our UV. So like I think it's like 99% of UV doesn't even make it past our lens. So the zoning out on the reflected UV, I think you're probably okay.
So a couple questions. Maybe I missed it. So your research so far has shown that the UV is not damaging the cells, right? So what has led to the recommendation then to avoid UV? Is it other parts of the eye or other studies or what?
Yes, if you want a really long-winded answer. So UV can cause cells to mutate in ways we don't want them to. So there's a lot of other cells in your eye aside from just those retinal pigment epithelial cells. UV, even just on our skin cells, can cause issues, obviously. Within our eyes, there are molecules that are highly reactive and can result in the breakdown of like key cellular function. And UV has been shown to trigger those molecules in accumulating, if that makes sense.
So I guess to that point then, you mentioned that you're running at about half the level of bright sunlight.
Right.
Is it, I mean, I understand the duration is long, but could it be where you need to reach a certain energy threshold then to
Yep.
cause
Yeah,
the breakdown?
for sure. And this project is like brand new. So I'm currently like in the depths of the trial and error of trying to get things sorted.
And I think the other part with all of these diseases is it's not just one extreme exposure to UV. It's all these other things coming together at once. So it could be one cell that was damaged by UV, and then it got a second dose of something else, and then we start the process.
I was wondering, so you showed some data that looked like it was from another piece of literature that was a similar cell line to the cell line you use, right? Where it's like that, I don't remember what it was called.
ARP19.
Yeah. How is that cell line different than the cell line that you're using from the pig?
Yeah, so we'll get there. The ARPE19 cells were originally pulled from a human donor's eyes. So they started out looking like the cells on the right. They have been transformed into something called an immortalized cell line, which is a really common cell line that's used in research because they're really easy to work with. Your question earlier about like, can the cells divide and all of that, that's one big pro to these immortalized cell lines is you can just keep growing them and letting them divide, transferring them to a new dish, and continue using them. So that's one big difference. The other difference is their shape is different, right? So the cells on the left have an elongated look to them. They don't have the pigment that's in the cells that we pull from the pig eyes. And so all of those things, although the ARP19 cells are retinal cells, they're different enough that it's just not super mimetic of what's happening in our eyes.
Got a lot of questions. So, question for both of you guys. How did you guys get involved in eyes? Because Sophia, you said your first and second year at undergraduate. To me, that means freshman and sophomore, yeah?
Yep.
Okay. And same thing for you, Dr. Vargis, like, because you went to Berkeley, right? Because California.
Oh, you're the woo.
I went to UC Davis.
Nice.
Yep. And, but like, even in undergraduate, I was not any of that. So why eyes, and how did you get down that path?
So eyes haven't always been my passion. It just happened to be that that's what worked out. The reason that I have been so involved, honestly, is I have two older siblings that, I watched go through college and then grad school. So, I was in middle school when they were in college. So I was just made aware of the opportunities. They were both really involved. So once I was in high school, I loved science and I loved math. And my favorite classes were when we got to do hands-on lab stuff. So I looked up the Biological Engineering Department here, stumbled upon Dr. Vargis. The eyes came later. That was just, it worked out that way.
So I started doing undergraduate research, a TA in a class put up before the class started. My PI is hiring 5 people, email, and then before I could talk myself out of it, I did that. But I started as a junior, so this was not this. And I did cancer biology, so we had an animal facility with rats, so I did that. So it was breast cancer research. I did patient research as for parts of my PhD. And I thought it was there were too many variables. There were lives with the animals. There were people that I had to talk to. And I thought, I want to do something totally different when I am a postdoc. And so I joined this group, and they had this microfluidic, this different way of growing cells. And they were working with somebody who was an ophthalmologist. And I convinced myself at the time that the eye was a very simple structure and it would be super easy to work on. I was wrong. It's super complicated, but here we are. Yeah, I've stuck with it more than the animals and the humans. Yeah.
Maybe one more question before we wrap it up? Anybody? There are no dumb questions. I'm just kidding, there are dumb questions, but you can ask one.
So this is a little bit unrelated, but it's like I'm a first year plant science major, and so I've been wanting to get into actually doing undergraduate research, and so what would you guys' recommendations for finding a way to get into that?
There's so many options. If you're interested in doing like specifically plant science research, I would start by looking at your department's website. I think all departments have a pretty detailed list of faculty, and usually they have some sort of summary of what work they're doing. The really nice thing about USU is that undergraduate research is a big thing here, and so the professors here want to work with undergraduates. So I know it can feel scary, but honestly, just sending an email to a professor that you're interested in their work and, read a little bit about what they do, introduce yourself, see if they have time to talk with you and if you can potentially get involved. I'd say like 9 times out of 10, you're gonna get a very enthusiastic response. So that's my student take. Do you have a different take?
No, I have the same take. I mean, my background is really like a top school and a high research environment and a national lab. And I love the environment at Utah State because we're so undergraduate focused. And so my weekly lab meetings are open. So if you ever have an interest in eyes, please come. They're on Thursdays at 1:30 p.m. But every semester they're open, and I know that a number of colleagues in my department have the same thing going for them. So just either look on the websites and figure out when those are and show up, send an email introducing yourself. And the other thing is that there are also the student research symposiums, so you can see what other students in your department might be doing in terms of undergraduate research, and then, tag along with that. Sophia does a lot of recruiting just by going out and talking about the research, and then 3 new students show up at the next weekly meeting, and it's because she's talked to them about exactly what she does.
I'd like to add an answer as well, because
Yes.
as associate dean, I get in our college, I get a lot of questions from undergraduates about that same issue, and I agree with these answers. Another thing I'd encourage you to do is to go to the office hours in the classes that you like and talk to the faculty members, whether they're lecturers or professors with research programs, and just ask them about others in the department too, and who's actively doing research on what areas. And that can give you a good insight from an insider's perspective. Well, thank you all for coming, and there are refreshments outside as well as lots of tables of activities, so please have a cookie, have some lemonade, and let's thank our speakers one more time.